Earth is home to an estimated 8.7 million species, but how do we make sense of that chaos? Why do we call a cat Phillies catus in London, Tokyo, and New York? Welcome back to the channel. Today we are diving into the diversity of life, specifically topic A1.3 of the IB Biology syllabus. This is where we learn how to map the entire history of living things. Biology is a constant balance between unity and diversity. Unity is what all life shares, like DNA and cell theory, while diversity is the incredible variation caused by billions of years of evolution. This chapter focuses on taxonomy, which is the science of naming and classifying that diversity so we can understand how every organism is related. The foundation of the system is the binomial system, popularized by Carl Linnaeus. Before this, scientists used long, messy Latin descriptions that varied by religion. Linnaeus simplified it to just two names. Take the grey wolf, Canis lupus. The first name Canis is the genus and the second name Lupus is the species. In your IB exams, you have to follow the rules of binomial nomenclature strictly. The genus must be capitalized and the species is lowercase. And the whole name must be written in italics when typed or underlined when handwritten. Skipping these formatting rules is an easy way to lose marks. We organize these names into a hierarchy of taxa, which acts like a biological address. It starts at the broadest level with the domain and moves down through kingdom, phylum, class, order, family, genus, and finally the most specific level, the species. As you move down from domain towards species, the organisms share a more recent common ancestor. For a long time, we thought life was just split between prokaryotes and eukaryotes, but molecular biology changed that view. We now recognize three distinct domains: bacteria, archaea, and eukaryota. Bacteria are your classic prokaryotes with no nucleus. Archaea often look like bacteria and live in extreme environments, but the DNA machinery is actually more similar to ours. Eukaryota includes everything with a nucleus, from plants and fungi to animals like us. The IB frequently asks how we distinguish between these, and the secret lies in sequencing ribosomal RNA. Deciding who belongs in which group has also evolved. Old school biology relied on morphology or physical shape. However, looks can be deceiving because of convergent evolution. For example, a dolphin and a shark both have fins, but a dolphin is actually more closely related to a cow than to a shark. To fix these mistakes, we now use cladistics. We build cladograms based on clades, which are groups of organisms that evolved from one common ancestor. Instead of looking at outward appearances, we look at molecular clocks. By comparing the base sequences of DNA or the amino acid sequences of proteins, we can see exactly how much time has passed since two species diverged. The fewer the differences in the genetic code, the more recently they shared an ancestor. Sometimes this DNA evidence is so strong it forces us to admit our previous classifications were wrong, leading to reclassifications. The most famous example in the syllabus is the figwort plant family. For years, these plants were grouped together because their flowers look similar, but DNA sequences proved they actually belonged to 5 different clades. Finally, to identify an unknown organism in the field, we use a dichotomous key. This is a simple but effective tool consisting of series of numbered yes or no choices that lead you to the correct species name. To summarize the core of A1.3, remember the binomials give us a universal language. The hierarchy of taxa organizes life from a broad to specific, and cladistics uses molecular data to show us the truth evolutionary history of our planet. Classification isn't just about labels, it is about understanding the 3.5 billion year old story of life on Earth. Thanks for watching and stay tuned for our next session on cell structure. Thank you.